Hello. Hello, good morning. Have you tried connecting? Projecting, everything is fine? Okay, I think a couple of people are still at breakfast, but we can start slowly. In the morning session, we have 2 talks introducing single cell paradigm, then our experiment that went into a paper you have all seen, hopefully by now, and then introduction from Alon Klein with a broad view of the field. This morning session is going to be recorded, so if you're going to yell insults or throw tomatoes or something, just be aware. And we're going to make it available as a video for you to go back to, or for people who missed the session. So I'm just going to let James deliver his talk. The only thing I want to say as a way of introduction, I remember James interviewing to join Kirshner Lab, and it seemed like it's been yesterday, and I asked him what is he interested to do. He said he wants to understand development at the single cell level, and I was laughing at his face. This is crazy, it's not going to happen for another century, I thought, and here we are. So.
Can everyone hear me? Yep. Okay, I'm just going to add one more thing to my introduction. By way of preface, I just want you all to know that I had a bit of a travel disaster last night, and so I've slept for just 1 hour. So forgive me if some of the things I say don't make sense. I'm sure they do in my mind. The words just probably aren't coming out right. But okay, so I was asked to give a talk kind of presenting the paper as a way of kicking off the Jamboree. But because I understand all of you have a copy of the paper and can read it and can get whatever details you like from the paper, in this talk, I'm going to de-emphasize detail and kind of frame it less as a presentation of what we found and more of kind of a primer for what I hope comes out of the Jamboree, highlighting in particular places where there's remaining uncertainty or there's additional opportunities, I hope, with our collective attention to learn more from the data that we now have access to. So my talk today is going to be in 3 parts. Part 1 is really just a story about how the first experiment with the data that you see in the paper came to be. Like all good stories, there was excitement, complications, heroes, bravery, heartbreak. It was all in there. Then I'm going to have a part in the middle of the talk explaining at a high level what we attempted to do in the paper by building a cell atlas and what it means and some of the things that we're going to explore doing together over the course of the Jamboree. And then finally, in the last part of the talk, I don't profess to have any answers, but I'm going to give you some short vignettes of ways we've attempted to use this new type of data to maybe learn new principles about development less to like be impressive than to stimulate conversation and encourage everyone else to like look for other ways we might learn something new over the course of the week. Okay, so the story starts in early 2015. As Leon said, I was just interviewing with the Kirshner lab. I was interested in studying cell differentiation. I thought single cell methods were pretty cool. And at the time, Alon Klein was still a postdoc in Mark's lab. And InDrops was actually not working, which is funny for me now to reflect on. But during my rotation, Alon did this massive experiment, got back some data, and it was a complete failure. Like, nothing worked at all. And I was like, oh no, I've just joined this lab. I'm going to do a thesis about single cell development, and the method doesn't work. So it was like a moment of nervousness there. But of course, Alon, being who he is, he worked out what was wrong, and the method came out just a few months later and works beautifully. The kind of like overarching concept of like my thesis project, like from the beginning, before we even had any data, was that we wanted to ask kind of what development looks like when considered over many genes simultaneously. Most of the tools that we'd had up to now, like studying cell fate transitions, hierarchies of states, and even like how many states there are in the embryo, relied on looking at one or a few genes at a time with high spatiotemporal resolution, or doing the whole genome, but then you're doing bulk samples, you have a mixture of populations, and it's hard to resolve like decision points, for example, between competing fates. So Alon had InDrops working. This was our general interest. I was a new PhD student, and so enthusiastically opened the Bible of Xenopus methods, and like was happy to see that there was a protocol. There was in fact multiple protocols to dissociate Xenopus embryos to single cells. Sounded pretty easy. Just follow the protocol, inject them into the microfluidic device, and out will come beautiful single cell gene expression data, and we can start to explore the questions that I was interested in. However, this is one of the first movies that I ever took, and I hope that one does not need to be an expert in microfluidics to understand that this is very wrong. There's long tendrils of DNA everywhere. The cells are stuck. Like, this blockage happened in 4 seconds. So it was just a complete disaster. Just like to give you a reference point, good data looks like this, where the left peak is your background signal and the right peak is the signal on your cells. My data looked like this. It was inverted. You're meant to have more than 80% of your data coming from the cells. I had more than 80% of the data coming from the background. So it was a bit discouraging, but we kind of broke the problem down and made some progress. But even after 6 months, I still wasn't getting the kind of quality data that we were getting in other systems. And a kind of real turning point in this whole project actually was when Alon decided that we'd spent 6 months trying to do Xenopus. It wasn't working so well. Another postdoc in the lab was doing the same approach in zebrafish. And Alon said, look, maybe James has spent enough time doing Xenopus, and he should just do his project in zebrafish. This made Mark so mad that over Christmas in 2015, he, Alon, and Leon were in the lab while I was in Australia visiting my family. And as you can see here, they had the embryos out. They were looking down dissecting microscopes. I don't even know what's going on with the horizontal microscope there. But they were trying a whole lot of things. And although they didn't completely solve the dissociation process, they really made some headway and reinvigorated the whole approach. And the two single most important things that came out of that Christmas was like being more experienced scientists than me. They put in place really good benchmarking assays to measure how well the dissociation was performing. So they were using DAPI staining to measure the viability of the cells you got out, and using ribogreen reagents to measure how much lysis was occurring during dissociation. This sets you up then to like systematically look at different conditions and measure like really how well you're doing, and kind of take like a increasing path towards like the protocol that ultimately gives you the data you want. So
James, quick
yep.
question. Which stage were you trying to do this
Yeah, so
Was it later on?
yeah, we were starting around gastrula stages.
At that time, did you maybe take age 22, 28, much older embryos that see whether that would work?
We were kind of trying a lot of stuff. Maybe we threw in some stage 22 ones, but the focus was mostly like late gastrulation. The thinking was that like, you know, once you get too far in development, the cells are really stuck together and dissociation gets harder. Too early, the cells are too big, they're too fragile, so it's also harder. We were like looking for the sweet spot in the middle where the cells aren't too stuck together, they're not too big. So we thought that was the best place to look, so yeah, that's where we were starting. But yeah, so after the PI intervention that saved single-cell embryology in Xenopus, there was about another 4 months worth of work, but it was all worth it because we found a set of conditions where you could do these experiments and have beautiful operating parameters like this, where a cell's just flown through and gotten captured with a bead. There's another being captured with a bead. You can look at these things being collected, and you have nice, round, stable drops. There's huge Xenopus cells in the droplets with beads. And going back to our original kind of like background versus cell signal histograms, our data was now in the range where we wanted it to be to move forward. So moving on to part 2, we have our data that's now of the quality that we wanted it. The first thing that we did was kind of what was most familiar in single-cell analysis at the time, which was just to take each time point separately, cluster it, and try and kind of make a catalog of all of the different cell states that are present in the embryo. So over the 8 time points that we profiled, in one day, this was like one huge experiment that took 24 hours and completely exhausted us. But we got these 8 time points, we clustered them. There was 202 cell states. It was already a result in itself. But if we wanted to study differentiation over time, we needed to have some kind of way of understanding how cells were moving from one state to another across time points.
Okay,
So,
James,
yep.
can you go back to the previous slide?
Yep.
For stage 8, is this only animal cap cell, or this is including cap and vegetable cells?
It's cap and vegetal cells.
Together?
Yep.
Ah,
Yeah. So
If you want to ask a question, raise your hand. I'll give you a microphone.
Related to this, because you had these problems with the breaking cells, did you check the representation that you really have the vegetal cells or then later on the endoderm cells presented as much as the rest? Because I imagine you lose those the most.
yeah. So I guess we had 2 metrics of that. The first one was by measuring how much RNA ends up in your dissociation buffer compared to how much is in your cells. You can estimate the fraction of the embryo that lysed during your dissociation, and that's where you lose cells. That was telling us that, like on a good run, like less than 5% of the embryo was lysing. And the second kind of piece of information we had was like if you take a histology slide of an embryo and you count the nuclei, and the huge like vegetal ones are endoderm, and then you kind of count their number relative to the animal cap cells, you get an estimate for how much should be endoderm. And that like ballpark agreed with the sampling that we were seeing in our data. We probably do lose slightly more endoderm, but it's like, so there is some bias there to be aware of, but we were at least representing each of the states. Yeah.
Speak up a little bit. The maximum, but still.
so why do you think the vegetal versus the animal difference does not show up here? And maternal including.
I see. So I guess you're referring to like veg T, for example?
Yeah, including some of those, yeah.
Yeah. So actually it does. You can see in the plot of stage 8 there, we've only annotated a single state, like all of the points are blue. But if you take that plot and you color it by veg T expression, you will notice that there is a gradient across the cluster. So one end will be higher for veg T than the other, and vice versa. It's just a decision that we made that the continua, like there wasn't a place where we could like cleanly fragment it with any kind of confidence, so it's just lumped together as one state. But you do see the asymmetries. Okay. Any more questions? Yeah.
And how many embryos did you use per stage, or is it as a single embryo dissociated and did the experiment with?
So for the early time points, we were using 10 embryos as input. By stage 22, because each embryo has more cells, we were down to about 5. In this first experiment, yeah. How many? Okay, so Mark's asking how many embryos we sequenced in a second experiment, and so I don't know if it was mentioned last night, but one of the exciting things we have to do at this Jamboree is we have a brand new data set that we got back a week ago. And we basically replicated this entire experiment and added some intermediate time points. And we have another 100,000 single cells to add to this analysis. And it's pretty raw. Like, we've taken an initial look at it, but there's surely much to be learned. And that's something that we should all enjoy doing together over the course of the week. But yeah, how many embryos? Again, it was like similar numbers. Like, we did another collection, maybe 10 embryos for the early time points, 5 embryos for the later time points. So yeah.
Going back to stage 8, a number of genes there, if you start looking where it's expressed, show only a few cells, 2, 4, 8, and I noticed it's likely because they're very low levels of expression. Does that present a big problem? Because I imagine it's not highly reproducible.
Can I just see if I understand the question? You're saying when you look at like certain genes on one of these plots, you might see just a couple of cells have detected expression?
I'm sure of that. I looked at a bunch, right?
Yeah, so that's definitely the case. And there are genes that are lowly expressed that we do see just a handful of detected transcripts. And from that, we have very little information about what it might be contributing to the cell population structure.
So does that mean you probably want to specify a limit at which the expression should be detected? Otherwise, right? Does
what would the limit?
For example, it has to be, okay, these numbers are just off my head, let's say more than 3 molecules per cell or more than 300, I don't know.
So it is true that when we're doing the analysis to like make these plots, for example, we impose like a minimum expression level to use that gene to learn about like how the cell population structure. We do have a threshold there, but when we then go back and look at where genes are expressed, we want you to be able to see everything. So like you'll be able to click on a gene that's in 4 cells and you can just see it. But it is an important point to know that like that is noisy information, and so you should interpret it kind of carefully. Yeah. Okay. So we have all of these cell states. We have single cell transcriptome information about them. But what we really wanted to understand was how differentiation is happening, how fate choices are being made across the entire embryo. So the approach that we pursued was to take like each of our time points is kind of essentially like a snapshot of the embryo at a particular developmental stage. Over time, these states are connected to one another. So we aim to connect like each different cluster to a parent cluster at the previous time point and build a scaffold. So I'm going to totally skip the details of how we did that today, but later in the week, we're going to do like a MATLAB exercise where we're going to try and walk you through reproducing that mapping between 2 different time points, and we'll go through the details of the actual algorithm there. But I'm going to jump straight to the output, which is what we're calling a cell state tree. So the cell state tree looks something like this. Every node is a cell state, and there's edges across time points reflecting these similarity relationships. Some general features of the tree are, although there was 202 cell states at each of the time points, there's only 72 distinct annotations. And that's because each cell state persists on average for 3.3 time points. So like, for example, you see The notochord from stage 12 through to stage 22, it's just the notochord. That's one distinct annotation, even though there's 5 states. There's 72 annotations. Of those, we could match 50 directly to XenBase as like a known cell state in the embryo. There was 22 where we could not like kind of unambiguously say this is like cell type X on XenBase. So in that case, we gave each of the cell states a name where it's like a name, a hyphen, and then a couple of the marker genes that defined that state. So I'm highlighting that now because I could not identify them, Caleb could not identify them, like and together we could not, but each of you are experts in different aspects of development, and it'd be really great if you could pay particular attention to these states that have hyphenated names and kind of see if it makes more sense to you than it may be made to us, and then we can, you know, interpret the tree better. Yep, and then there's 28 branch points on the tree. Okay. Yep.
So you mentioned that this is a new data set. Do you think that data set will change something in yourself? Oh, sorry. You said that you got additional data now from a separate experiment. Do you think that this tree will be much majorly altered? Will we get more cell states defined? Or do you think they will just confirm what you found already?
So I can...
You haven't analyzed the data yet in detail.
So we have, and the way we've analyzed it, it's not changed the structure of the tree. But I'm hesitating to answer you because the way we analyzed it is to scaffold the new data onto this analysis. So we weren't exactly testing this with a view to falsifying it. We were just grafting new data onto it. And with the new data, there's some satisfying results, like you can split some clusters, particularly small clusters now, into multiple new subtypes. So there's additional nodes on the tree. But yeah, in terms of like changing the parent assignments, we weren't really, well, to date, we haven't analyzed that data set in a way that it would be possible to do that. Yeah.
Remember, there is also additional time point.
Oh, you have also additional time points. What are the complete replication of existing experiment?
Yeah, so there's a complete replication of these time points. And then there's also, we added stage 11 and stage 13. The reason being, as you go from stage 10 to 12 and from 12 to 14, there's just so many new cell states appearing. And we knew from like XenBase that we were missing some intermediates. And we wanted to try and sample those. Yep.
I would like to ask about what you call the like cell type. So is that the algorithm assigning a cell type name, or you are doing it like manually?
So the names are a totally manual process. We're chopping up the data into clusters using clustering approaches, and there's a million of them. And you just kind of choose one, and you can, like in our case, we compared it to a separate one, and you show that the structure is robust. But in terms of interpreting what it is, basically you take a cluster, you ask what genes are specific to it, and then you go to XenBase in our case, and you say where are these genes, like what cell types do they define?
Well-known markers.
Yeah, exactly.
James, can you turn up the mic?
I'm maxed out. I can try it right.
We need to turn up James.
So I have a question. So the cell state seem to be less than the cell types, right? Related to the question. So then is basically depending on how you set the cutoff rate, the threshold. So so because like for example stage 8, you only have one cell state. Well, we know there are different cell types. So if you twist the threshold, the cutoff, do you, I mean, have you played with the parameters to see whether you get more cell state versus cell type match?
Yeah, so without going into cell, is that better? I'm choking myself a bit, but you can hear me, so let's go with it. So I don't want to get into cell type versus cell state right now because it's like a philosophical question, and we can talk about that over beer later. But in terms of changing your parameters to change the number of clusters you have, yeah, I mean, you can arbitrarily make it so strict in defining a cluster that every cell is its own cluster. And then when we sequence 10,000 cells, we have 10,000 states. But it's total nonsense. Like, there's no reliable differences between some of those cells. So later in the week, we're going to do a MATLAB tutorial where I'm going to walk you through taking one of these time points and doing the clustering for yourself. And so you'll be able to explore on your own what changing the parameters does to like the number of cell states that you see and what defines them. But basically the heuristic is you're wanting to cluster the data as much as possible such that two different clusters really are different to one another. If you over-fragment the data, then you're going to have one state that you've just arbitrarily chopped in two. There's not going to be real differences between them, and we don't want that in our analysis, so you're trying to find the sweet spot between those. And the truth is that process is really manual. So it's judgment calls by myself and Caleb as we do the analysis. Yeah.
Just one thing. We stopped at stage 22 for a couple of reasons. One is that there was enough to analyze, but the other was that the dissociation methods that we've finally worked out are purely chemical methods that don't involve any enzymatic treatment. And they do dissociate, whether there's some lysis in the apparatus at some low level that we've talked about, but they do dissociate the cells 100%, including the pigmented epithelium completely dissociates the single cells. Past stage 22, that method, it briefly tried, isn't as completely effective, and I suspect requires some enzymatic treatment as well as some of the physical treatment. So, that's the point. I quit. It was effective enough. So I think that should people be interested in further stages, they would need to put a little more time into working out better treatment. And I'm hoping that we can continue the Slack approach that we have here so that people could post even modest improvements. So for those people who are interested in later stages, they can make faster progress. Okay.
So we've had some good discussion in terms of like understanding what this tree means. There's two reasons a tree like this is useful. One is that it gives you a kind of interpretable view of what the data looks like, all in one summary. Like you can understand each of the branch points, each of the cell state transitions at a pretty granular level looking at this. The second reason it's useful is you can simply use it to chop up the massive data set that we have and like zoom in and look at a particular group of states that are related to each other in a meaningful way. So for example, I think a lot of what will be happening this week using the tools that Caleb's built into like the online browsers for this data is here we take the subtree rooted at the intermediate mesoderm at stage 14. So we're taking all of the cell states that come from stage 14 intermediate mesoderm. We're taking those single cells out and we're making a representation of those. That's what we're looking at here. So that over time you can see cells like kind of flowing into these 5 different endpoints, and then you're free to like. So a common goal of like when you're studying a fate choice is to identify regulators that are associated with one fate versus another. It's readily accessible once you have a representation like this, just to ask which genes are correlated with moving down each branch. Here I'm just showing like some known ones, but if you like take each of the choices that have occurred, you can make heat maps of all of the different genes that are going up and down as you make each decision. And this is kind of like a resource that becomes accessible from this tree. Yep.
Thanks. James, yeah. For this unity tree, do you see any common precursors for neural and mesoderm? Because in mouse and in fish, there's this posterior mesoderm precursor cells that give rise to both lineages. But from your tree, it looks like there's no connection of these two. So is this something unique to frog, or maybe it's missing from the data?
So this, I believe the state you're talking about is the tail bud?
Yeah. In some sense, yes. Yeah.
Yeah. So there is this tail bud state on this tree. It is, where is it? Here we go. It's this one. This is the tail bud. So the first part of your question, do we see a state like this that's a mixture of ectoderm and mesoderm? We do. This tail bud state, it expresses like SOX2, which is a transcription factor of the ectoderm. It also expresses brachyury, which is a transcription factor of the mesoderm. So it is this population of like mixed developmental potentials. Our tree does not tell any story about the tail bud making different tissues. I actually don't know whether the tail bud's meant to have been producing neurons and mesoderm over the timescale of this experiment, and we haven't detected it with our tree building approach, or if maybe it's going to do that later in development. But yeah, that's an interesting thing to explore. The tail bud's there, but we've not tracked any of its progeny. Mm-hmm. Yep.
Were you able to fit all the cells into this tree? Is it comprehensive? Did you fit all the cells into this tree, or are there some cells that didn't just fit your clustering and you excluded from this?
Yeah, so all of the cells that we assigned a state are in this tree. However, as we analyzed each state individually, there was two criteria by which we excluded cells. One was if we thought that they were background. So the characteristics of background was they lowly expressed everything. We like believe that's a technical effect, so we got rid of those cells. The other thing that we excluded was what we believe are to be doublets. So doublets occur when two cells get co-encapsulated in a drop. They get the same barcode, but actually there's two cells. So doublet states, they're rare, first of all, in line with like Poisson statistics. And they also express markers of like two other like readily identifiable types. We excluded those as well. They're not in the tree. However, in the raw data, all of those cells are present and they just have a cluster label of like -1. They're excluded. So it's possible that with like some sleuthing through them, you could discover some stuff that's like rare cell states that we just like, well, I don't know what that is, and got rid of it. Yeah.
There are also cells with cluster.
Oh, yeah. So the other thing that's excluded is a cluster had to have at least 10 cells. So anything that had less than 10 cells in our data never forms a cluster and never makes it into the tree. Yeah.
I wanted to add a quick addendum to that, which is that the germline is in this category of things that are not in the tree, and so there may be like profound omissions from the tree. Because
We found germline cells.
Right.
Those are not on the
They're not on the tree, so that's an example of a cluster that was too small.
Let's
Yeah.
speak into the microphone again.
How do you deal with pluripotency states? Do you recognize them at all?
So pluripotency, in my view, is like a function. It says that a cell can generate progeny states of all three germ layers. We're measuring gene expression states, so like no, we're not really directly assessing pluripotency.
I would disagree. The tail bud is pluripotent.
Okay, so the tail bud is pluripotent. I agree. Well, I mean, papers agree.
Is it bipotent or pluripotent?
Can we leave this exciting debate in the lecture?
Yeah. So my point was only like, sure, we can see the tail bud and we can see its gene expression is mixed between germ layers, but it could have been the case that the tail bud was not pluripotent at all and it had mixed gene expression. Like our data doesn't directly speak to the potency of a cell. Yeah. I'm out. Okay. let's press on. So the final footnote I wanted to make in this section about building the cell state tree is that this map that we're like holding up and saying is useful, it's just one way of looking at the data. There are surely going to be other ways of looking at the data, potentially better ways in the future. I mean, in our lab alone, Dan Wagner, who's the postdoc who was working on zebrafish, came up with his own independent way of looking at the data. And that built like a KNN graph of the entire data set all at once. And you get applied to the Xenopus data set, you get a picture like this. It's clearly different to the cell state tree that I showed you just before. Like it's got its own unique kind of utilities, perhaps some drawbacks. I just want, I want to emphasize the point that as we're exploring this data structure, it's just one way of looking at the tree. So like if there's things about it you don't like, feel free to speak out. Like there's probably a better way of doing it, and like finding the pitfalls is a useful exercise.
Not at this moment though, we will have ample opportunity to discuss what this representation means, how to use it and so forth.
Yeah, okay, so I've said there's many ways to do it, as might not be the best, but there is some like underlying kind of truth to what's going on. Like so on this plot on the right, we're showing the edges on our cell state tree overlaid on the graph that's been built by this different method. And you can see there are some areas like this wiggle's pretty weird. I don't know what's going on here exactly, but overall there's good qualitative agreement. Yeah. Okay, so part
the temporal change and
okay, sorry, yeah. So the colors here mean time point. The blue being stage 8, walking through red is stage 22. It's the same time points in the previous data set. And so what we're looking at is cells radiating out from like the center, which is the stage 8 gene expression state, like kind of fanning out into these multiple different expression states over time.
And so the fanning out means they're actually getting farther away from each other in terms of similarity, right?
They're progressing away from their state at stage 8, yeah. Yeah. Okay, so now the hardest part of the talk, which is kind of, at the start of this project, there was like this kind of view that if we could look at development over many genes at single cell resolution, we'd really be able to say something, like learn some principles. And like my honest kind of feeling about this is a little bit dissatisfied. I don't feel like we really delivered any kind of crisp new concept about development. But I still think it's a worthwhile thing to kind of shoot for. So I hope over the course of the week we can kind of look for these types of things. And in this part of the talk, I'm going to give like 4 very short vignettes about attempts to use kind of unique features of the data to learn a principle about development. And then kind of that can be food for thought over the course of the week. Okay, so vignette number 1 was testing the developmental origin of the neural crest. This was kind of an interesting question to approach, in large part because Carol LeBon and her group had recently published a paper suggesting that the neural crest emerges through a new pathway to what was classically thought. So classically, which is model 2 here, it was thought that the pluripotent blastular cells commit to each of the different germ layers, and then the neural crest differentiates from the ectoderm, in particular at the boundary between neural and non-neural ectoderm, and it kind of regains pluripotency. So the idea was that there would have to be some kind of independent pluripotency network that's coming on in the neural crest to make it able to make fates of the 3 germ layers. LeBon and colleagues argued on the basis of kind of some shared marker genes between the blastular cells and the neural crest cells, and the fact that these genes were important to pluripotency of the blastular cells and of the neural crest cells, that there was a population of cells that kind of retained pluripotency from the blastula. So there was not this like transition away from pluripotency and then back to pluripotency. It was just kind of a continuous progression where there's a state set aside in the blastula that stays pluripotent. Our data, at least in the cell state tree, like immediately kind of challenged that kind of view to us, because there was certainly no like separable gene expression state that connected the neural crest, which at the time we were detecting at stage 14, to the stage 8 blastula cells. They were clearly being assigned to neuroectoderm as their parent cell state cluster. And if we cut out the single cells along this trajectory and at the neural plate border, and we kind of tracked what was happening over time, there didn't seem to be like the hallmarks of like a state that was set aside and like uniquely contributing to the neural crest. And so we were kind of confused like as to how Le Bon had like created this entirely different picture to the classical model, and then our data again was providing a different view. And so you can kind of ask like why do our conclusions, which support model 2, differ to what Le Bon said? And so in terms of like a concept with looking at this data, like one of the things that makes it useful is you're looking at lots of different genes and you're looking at them quantitatively. So we went back to like this differentiation into neural crest, and we took the very genes that were used in the paper by Le Bon to argue that there was this continuous state. And so what we're looking at here is gene expression over time. Each row is a gene, and the x-axis is time. And you're looking at the kind of the dynamics of gene expression. And so broadly speaking, there's a lot of dynamics happening. There's many genes going down, many genes coming up. There's identifiable intermediate states. This is like, first of all, not really consistent with the view that there's a population set aside and is staying pluripotent as it's coming to neural crest. But then even more troublingly for the model by Le Bon is if you look at the genes that they had provided in situ evidence were expressed in both tissues, they themselves are highly dynamic. Like in general, most of them were either going down tenfold or up tenfold, and it wasn't like steady expression of these genes over time. Maybe if you like develop your in situ more, you can detect some expression, but it's not a stable marker of like a state that's set aside from the blastula. And then second, if you look.
Just a quick question about this. Isn't it a general assumption that any given gene could be very dynamic, so it does not necessarily be a lineage marker, so it's all the combination of things, right?
Yeah, I think, yeah, in general that's true, but like the title of the paper by Le Bon is like shared regulatory programs suggest retention of blastula stage pluripotency. So if the whole foundation of your argument is these shared regulatory programs, and the regulatory programs are like tenfold dynamic, like up or down, it's starting to look like a bit of a strange argument. Like I said before, pluripotency is a function, and it's not necessarily completely encoded in the gene expression in a way we can identify. So it might still be true that there is like a subset of cells that are pluripotent, but the gene expression just like it's not consistent with this idea of a like continuous state separate like from the blastula to neural crest. Yeah. Yeah. So that's yeah.
Could it be just a very subset of a program that is required for pluripotency? Then this subset of program is sort of buried in vast of other gene expression patterns.
Yeah, so it's entirely possible that we detect all of these genes that are going up and they're going down, and in fact there's just like a small group of genes that stay on and make it pluripotent, and that would be enough for the view that like pluripotency is retained from blastula to neural crest. The problem is the genes that were proposed to fulfill that role are themselves in this group of really dynamic genes. They're not just stably on defining some population. They're going down tenfold or they're coming up tenfold. And then moreover, the same genes that are kind of functionally tested in the paper are not specific to the neural crest or the blastula. Like, so SNE1 was one of the genes that was knocked out and shown to be important for pluripotency in both the blastula and the neural crest. Sure, it's on in the neural crest and contributes to its pluripotency there, but it's also expressed elsewhere. So it's not sufficient for pluripotency, if that makes sense. So just like that, I wouldn't say that this is like, yeah.
So why is it when you do in situ, that SNEL comes on, behind your ears, but it doesn't come on in all these other places? Is the in situ data just not linear enough, or what?
It does come on there.
As much as that, because people use it, at those stages.
Not on a request specifically. Oh, I've done it.
Please raise your hand and get the mic if you.
Yeah, so in situs actually do support like that SNE1's on in like the marginal zone, for example, and some mesoderm. Yeah. So yeah, like I said.
It's a little better, but it's still not specific.
Yeah. So like I said, I don't think that this is necessarily definitive proof that the neural crest isn't like a pluripotent lineage from the blastula, but it sure does look weird with the model that was proposed, that these genes are kind of stay on in a subset of cells and make them pluripotent. Yeah. If your model starts to look this weird, like I think it's time to like doubt it a little bit.
Then from your sequencing data, can you derive, single cell data, can you derive something that is unique about the pluripotency in neural crest? Or not get to that level yet?
There is a whole world of people who care about this, namely like stem cell biologists, and there's been attempts to do things like this with algorithms like CellNet, etc. They like take a bunch of gene expression states, they aim to predict pluripotency based on the gene expression profile. Like, I'm just saying this, people think about it, like I have not thought about it.
Didn't you say also that you lost or excluded cells that didn't fit to a certain cluster or that formed a cluster that were less than 10 cells? And couldn't you imagine that pluripotency genes would be some cells that do not really belong anywhere due to the transcription profile and that you just excluded them?
Yep, so absolutely. But we can put bounds on how rare that state would have to be. And it would have to be rarer than 0.2% of the cells in the embryo to escape our previous analysis. And with the new cells that we've added, it would have to be like much rarer still, and we still don't detect this state. So yeah, if you want to argue that these genes can be tenfold dynamic, they can not be specific to the neural crest, and they can mark a population that's like less than 0.02% of the embryo, and that defines the pluripotency of the neural crest, you're free to do so. But yeah.
Your data does clearly show that looking at the expression in more detail doesn't fit Carol's model very well. Nancy Papalopoulou has done work showing that oscillations are very important for lots of cell state decisions. And you wouldn't detect an oscillatory kind of group of cells with this analysis. Not to keep defending this when for any particular reason, but just as you pointed out, it could be what's going on.
Yep, can't rule it out.
You can rule it out based on in situ hybridization analysis.
I think that's really a good point. And I think it's a good question. Maybe we should ask ourselves whether we could detect oscillatory groups. In particular, if the whole transcriptome oscillates, it might be difficult to detect it. But if there are just a few genes that oscillate, and if we were sensitive to that issue, could we detect it? I think that's an interesting question to raise.
Yep. Yeah. Actually, Caleb is a really good person to talk to if you want to try and detect oscillations in single cell data. I've actually thought about it a lot.
Who's this?
Caleb. Yeah.
I mean, I guess the question would be also in the somites would be to be looking for that. And maybe that's something we can figure out even at this meeting to focus on.
Yeah, just a quick comment about the stem cells and how rare they are. For example, in a mouse has been quantified that at the stage 10.5, 11, there's only 2 stem cells, true hematopoietic stem cells in the whole embryo. So they are rare, but probably in this kind of pluripotency is different. I don't know.
Cool.
Coming back to the issue of oscillations, I think probably the presemitic mesoderm would be a good one to look at because they're effectively they're oscillating, but in a synchronous manner. I think the work that Nancy does is actually looking at oscillations at the single cell level that are not synchronized. And I do wonder to what extent would whether this data would be able to find those. It would be really quite interesting. But
What about changes in gene expression according to the phase of the cell cycle the cells are in?
Another kind of oscillation.
I mean, you must be able to get that out of the data by looking at, correlation of transcripts for, I don't know.
Yeah, you can definitely detect cell cycle stages from gene expression data. Yeah.
But you haven't looked yet to see how that might influence.
Not in a detailed way, no.
I agree with that, but I should point out that most of the oscillations, certainly in the early embryo, are not transcriptional, but involve phosphorylation, protein degradation. So, at some point they will be linked to transcription. And it is true that the functional importance of the transcriptional oscillations is very unclear, even in yeast, where it does oscillate. They do oscillate with the cell cycle, but where if you put them on constitutively active promoters, there's no effect on the cell cycle. So I think we have to be a little careful, with a negative.
But presumably, if your cells are in G2, they're going to have like twice as much transcript. Is that true? Do they have different amounts of transcript for stuff when they've gone through S phase?
That's probably true that they are twice the size.
I think Ben's doing some work in this, and there's some really interesting stuff into that.
So for those who are interested in oscillations, there is a paper by Owens and on the temporal time expression data is up to 66 hours. And in the supplemental data sets of figure S6, we have seen expression genes that oscillates during this process, and there are several genes that oscillate. So it might be interesting to overlay the information and try to identify whether those genes are actually oscillate in your analysis.
Talking about the supplementary figures, I don't think they were made available to us, right? At least the PDF file of the manuscript and it contained it.
I'm more than happy to share them. Yeah.
I mean, by reading the paper on the plate, it would be nice to have that.
Yeah, I'll post it to the Slack as soon as I'm done with this talk. Yeah. So it sounds like there should be an oscillation group for the rest of the Jamboree.
Just shared on the Slack, but there was a really nice paper in Nature Cell Biology with E8.5 mouse single-cell data where they characterize the oscillation in the pre-Semitic mesoderm, so that might be a good model for us to compare to.
Okay.
I can use the second cube, but it will be broken.
Okay. So vignette number 2 is kind of a super ambitious thing to attempt, and like we're just going to scratch the surface now, but we do have access to from the efforts of Dan very, very similar data for zebrafish. And so one of the analyses presented in the paper involves a comparison of the two cell state trees. So one of the figure that introduces this comparison is here, where on the left we have all the Xenopus cell state tree, as you've just seen it, and on the right we have the same for zebrafish that's been constructed like through the same approaches. And they're interleaved such that orthologous cell states are connected in these shaded red groups. So you can kind of see cell states that are common to both developmental programs connected over time during early development. And the first point I want to make is this is just like a super fun thing to look at and explore. Like you can take the epidermis, for example, and here we've cut out this like constellation of states where you can see there's quite a big group of cells that are different between the two species. Then you can make spring plots of that group of cells in both species, and you can see that in Xenopus on the left you have your epidermis, it's making ionocytes. The same thing happens in zebrafish, you have epidermis making ionocytes, but then there's a bunch of states that are present in one species and not the other, like Xenopus has goblet cells far earlier than zebrafish. It has ciliated cells, presumably to distribute the mucus that's being produced, et cetera, et cetera. And you can detect all of these species-specific features. You can then look at the gene expression that's coming up in one versus another. So like this is just like, it can be done, no real principle, but I just wanted to share it. So I guess in terms of like looking for principles or trying to answer like new questions, the one thing that like was approached in this paper was asking like how conserved is tissue-specific gene expression between the frog and the fish. So when we say tissue-specific gene expression is conserved, what we mean is this. So here we're looking at SOX2. Every dot on this scatter plot is like one of the matched groups of tissues, like in these red shaded areas. And you can see they lie like scattered along a diagonal. The correlation's good, it's like 0.9. And indeed, like we know SOX2 is expressed in all the neural tissues in both of the species, like some of the early ectoderm in both species. This is a gene that we know is super important for a bunch of cell states, and it's in the same cell states across species. There are also genes that are really poorly conserved. So here the example is GATA5. When you do the same analysis for, yeah?
Let's finish your sentence.
Okay. So when you do the same analysis for GATA5, you can appreciate that it's expressed in erythropoietic cells only in Xenopus, not in zebrafish. And it's been shown that if you mutate GATA5 in Xenopus, it disrupts erythrocyte development. So here we have a transcription factor that's on in red blood cells in one species and is functionally required in that species, but is not at all present in red blood cell development in the other species. I think it's pretty interesting.
So James, just with this comparison between Tropicalis and zebrafish, you have of course the whole genome duplication that teleosts underwent. Did you take the whole genome duplication that occurred at the onset of teleost evolution, did you take this in account? So if you look at GATA5 expression in zebrafishes as GATA5A and B that you put together, or how carefully did you take care of this problem?
Can I defer answering this question to Leon? Leon is our gene matching wizard and deals with all of these complications. I don't know.
Drill down into this question, but probably outside of the format.
Yeah, it's super complicated. Yeah.
I mean, I know from my own experience looking at cross-species comparison.
Talk about the matching genes.
There may be a little follow-up thing here. You don't find all the cell states represented in both species. However, you mentioned that the cell state definition depends on the parameters that you use to basically the thresholds that you introduce, etc. Have you tried to play around with these thresholds to try to make them match up better?
To find missing cell states? The truth is not in a systematic way. We approached the problem, like the problem of clustering, the same in both species and had kind of like similar guiding principles. But there's not been a significant, like a systematic variation. It's like really hard.
Okay.
Caleb?
I just wanted to follow up that comment about the fact that in the figure, the red shading shows states that have been matched, and white background means we've not attempted to match them. But I just wanted to emphasize that simply because a state isn't matched doesn't mean that that we couldn't find any cells in the other embryo that corresponded to it. It just means basically the matching that we're showing here is our high confidence matching. So it's the one. So if we want to make statements that genes are conserved or not conserved, we want to be sure that we're comparing states that really are the same, because if we connect two states that are actually just different, then of course gene expression would appear to not be conserved. So just because something isn't matched in this figure or in the paper doesn't mean we couldn't find the corresponding cell in the other embryo.
So related, this poor correlation issue that we just discussed, could it be, for example, GATA5 in this case, the differences between fish and a frog is because some other GATA factors are replacing GATA5, such as 4 or 6, them having a very similar role. That we have seen, similar thing we have seen in other families, for example, between mice and human.
Yeah, so exploring why this is the case is a totally interesting question. And like to speak to your point, like another example I know is the neural crest. In Xenopus, there's like 3 SOX genes. I think it's like SOX 8, 9, and 10. And in zebrafish, there's also 3 SOX genes in the neural crest. It's like SOX 4, 6, and 10. And the 4 and 6 are completely not orthologs to 8 and 9. So there's this mysterious triplet of SOX factors in the neural crest in both species, but 2 of them have changed identity, as like it appears across the 2 species. And yeah, I think these puzzles are like super interesting.
Yeah.
And like it's like a subject for future exploration. Yeah.
I maybe can add on to that. I mean, that's a very important point that you always have to look at the paralogs. I mean, from our own experience with PAX 2, 5, and 8, you have a situation, for example, in the thyroid gland, you need PAX 8 for the development of the follicle cells. Now, in Xenopus, there's no PAX 8 expression in the thyroid gland, but it's PAX 2. So there was probably in early evolution, you had a state where all 3 or at least 2 of them were expressed, and then species select if you silenced one of the 2. Mm-hmm. So that's for me a very nice example where the paralogs basically have been separately selected after they diverged from each other, the mammalian line from the amphibian line. Mm-hmm. And in zebrafish, I can't remember the exact situation, but there's also small differences again. So one has to really also take the whole paralog situation into account to see whether the missing GATA here might be compensated by GATA 3 or 4. That might be the, I don't know what is the direct paralog here in this case.
Yeah, it sounds really interesting. Okay, so that's good correlation, bad correlation. Now we look at all of the genes that we can match between species, and you get a histogram that looks like this. And you can see that there is like detectable conservation of gene expression between the 2 species, but there are many genes that are like, at least like to my intuition, surprisingly poorly conserved, to the extent where like a marker gene for a tissue in one species in general performs poorly at marking the same tissue in the other species. And like here, I'm showing you a heat map where you're using just gene expression correlation between the matched cell states of both species. And if gene expression was perfectly conserved, we'd expect like a block of red along the diagonal. You can see that the picture is far from that. There's a lot of off-diagonal correlation. The correlation on the diagonal is quite weak. And so like this is kind of like an interesting observation to make, particularly like when like evo-devo biology like very commonly relies on using a marker gene for a certain tissue to look at a whole bunch of different species and study like how the tissue's changed, its location, its gene expression, etc. If we know now that the marker genes in general perform poorly between 2 species, you need to be really sure that your marker gene's marking the thing that you actually want whenever you're doing a kind of study like that.
I think we should all remember that zebrafish is a poor example to compare things with. The teleos have gone off in their own bizarre direction of partial genome duplication, and also radiated tremendously into very interesting species. I think more interesting, not that we have it available, would be to compare it to something like sturgeon, which is a more...
Sturgeon, the spotted gar the spotted
Spotted gar,
Did
or
that-
some more basal animal along the way. So I think we shouldn't. This may not turn out to be as serious a problem as it looks right now.
So we're aiming to this to end this not at 10 as scheduled, but let's say 10:20. So we have a few minutes to set up for a remote presentation from Alon. So let's hold the questions and let James wrap up.
Pretty close.
So 15 more minutes?
Yeah, I think we can just continue at the current pace. We'll get there.
Okay, good.
Yeah.
Might this analysis also be a sensitive way to find out which zebrafish genes are misannotated? Because a lot of them used to be misannotated in terms of the orthology.
Okay, so again, I refer to Leon. Leon has zero respect for any gene annotations. He makes his own on the basis of information that he finds to be more reliable. So yeah, it is a big problem.
So yeah, we'll have a separate discussion about which gene is what tomorrow, I think.
Okay, so if genes can vary a lot in how well conserved they are between the two species, you can ask what features of genes predict whether they are conserved or not. I mean, the first, like, and dumbest thing you can do is kind of like a gene ontology kind of analysis. You take your like highly conserved genes, you ask what Go terms are enriched in that fraction of genes. Not really particularly surprisingly, but somewhat reassuringly, when you do this, you find transcription factor associated gene ontology terms are associated with your conserved genes. So, you know, you can imagine that there's certain transcription factors that control your identity. They're conserved. The rest of your genes are a bit more flexible. They're more poorly correlated, whatever. But yeah, kind of just so. The more surprising observation was, like Caleb noticed, that your gene sequence similarity is entirely uncorrelated to your gene expression similarity, which is to say, like, it's counterintuitive to me, because consider two genes. They're perfectly conserved in sequence across evolution. They're doing something important. It stands to reason that you'd expect them to be like expressed in the same tissues perhaps, or maybe doing the same thing in different tissues. But the expected gene expression similarity is exactly the same on average as for genes that are 20% sequence similar.
I just wanted to add a clarifying point that we're only looking at genes that are already considered reciprocal best sequence hits. So obviously, if you just took any random pairs of genes, then there would be an enormous, as shown in the plot on the left, a strong correlation between sequence similarity and expression pattern similarity. But once you condition on the genes already being reciprocal best hits, then that association disappears. So once two genes are reciprocal best hits, it seems not to matter anymore how related they are in sequence.
So this means there is possibly a gene which is very similar, but not the best match by sequence, but the second best match by sequence, which would have exactly the same distribution across tissues.
Just wondering whether what this is really telling us, which is perhaps something that we already maybe knew, which is that most genes are not that dosage, you know, their activity or function is not that dosage dependent. There are dosage dependent genes, but I'm not sure that that's the majority.
But I think that this says more about reciprocal last hits than about this correlation. There may be a functional region of this gene which is extremely important, and it, as you say, it comes up number 2 on the blast thing, and it's not chosen. So I think before we get carried away by this, we have to really know in more depth how, what's similar and what's not so similar about it.
Okay, great. So like, let's treat this as like question opening rather than question answering for the purposes of this Jamboree. These are types of questions, and we're already in the weeds, which is great. Yeah.
How easy is to match the stages between Xenopus and zebrafish? I think that could be one of the problems, isn't it? That one stage you're calling Xenopus is absolutely different on a fish, or you don't know the equivalent in a fish, particularly at this stage.
Yeah.
Stages.
Yeah, so the matchings that you make between tissues absolutely affects the correlations that you measure. In fact, like we had early versions of tissue matchings that showed like even worse gene expression conservation between species. Once we improved the matching, we detected more strong gene expression conservation. The only thing that we've done to like check that our matching is like somewhat sensible is you can randomly permute the matchings and ask whether you improve the detected correlation, and no like reasonable substitutions improve the correlation by more than like 0.1 or something.
So James, I guess this issue you are discussing about is probably related to conservation of the coding sequence versus non-coding sequence, such as enhancer and regulatory regions.
This is protein sequence similarity.
Right, but then different tissue expression changes, probably more associated with non-coding changes.
Yep.
So probably those bifurcation of coding versus non-coding changes during evolution give you this kind of pattern.
Mm-hmm. Yep, that could be the cause. Okay, I'm just going to move on because we're running out of time. So the last two vignettes are examples of like attempts to find kind of a principle by just asking like what happens across every lineage in the embryo. Like anything that's kind of repeated across all of the fate choices like may be important is the idea. So one thing we looked at is multilineage priming. So I'm sure most people are familiar with the concept. It's where competing regulators of two different daughter states are co-expressed in progenitor cells. There's dynamical models where you can like describe the competition and show that it gives you nice fate choices that's satisfying. There's cool experiments where you can force co-expression of these competing regulators and show that it gives cells like the potency to make both of the daughter fates. Like you can even reprogram fibroblasts to IPS cells just by forcing competing regulators of different germ layers. Like, so it's like it's this decision-making motif that's also associated with kind of like a stemness. When we look for that in the single-cell data, you're looking at something like this. So here we have the fate choice between the neural plate and the dorsal marginal zone. And you can see that SOX2 is coming on in the neural tissue, brachyuria is coming on in the mesodermal tissue, but they're overlapping in a large fraction of cells at stage 10 before they're refined. So this is what we're talking about. The only like enticing thing that came out of this analysis is that there's a global trend where multilineage priming becomes much less frequent over time. So like at stage 8 and stage 10, you're involving in frog like 20 to 30% of your differentially expressed genes, and in fish it's like as high as 70% of the differentially expressed genes. It's going to almost 0 by the end. Please do not read too much into these like precise numbers because they depend on the sequencing depth. Like the deeper we sequence, the more multilineage priming we'll detect, just for statistical reasons. But the trend should not be sensitive to that. So there's like suggestion of something meaningful there where multilineage priming decreases over time. What it means, I don't know. But again, I'm just like throwing things at the wall as like ideas of what to go after. And then in a similar spirit, we can ask about how cell fates are specified. Like one idea is that there's like a single regulator that's coming on and making each different cell type. Another idea is that it's the combination of regulators that's giving you each cell type, and each transcription factor will be used in multiple different contexts. We can now tally that up over every lineage in the embryo and show that half of transcription factors over the time period we measured are in one tissue, but half are used in multiple tissues. And if you look at each fate choice over time, the like fraction of differentially expressed genes that differentially expressed transcription factors that have been used elsewhere already climbs to like 90%. So by stage 22, when you're making a new cell fate, 90% of the transcription factors you're turning on have already been used in some other fate choice. So again, it's just a trend. What it means, like, there's not really a particularly satisfying answer right now, but it's in the spirit of using the fact that we can now look at every lineage all at once. Okay. So yeah, that's it.
So there is coffee break served, and in less than 15 minutes we'll have remote presentations from Alon Klein.
James, may I have a question about the multi-lineage priming? So your observation seems to suggest that earlier the multi-lineage priming is a more prominent phenomenon than later stages. Could this be associated with the property of the earlier the embryos are much more plastic, and maybe they are just more prone to perturbation? For example, as I talked to Mark yesterday, cells, when they are dissociated, there's sometimes there can be significant changes, single cell versus a whole cap, either neural or epidermal. So when you do single cell sequencing, whether those kind of manipulation to dissociate cell will cause a change in some gene expression, at least in the early stage 8 or 9 or cells.
So, like, does it correlate with how plastic a cell state is? Like, these are the kinds of ideas I was thinking about as I did the analysis. To test that would be awesome. Like, you'd need to somehow control the extent of multi-lineage priming and measure the plasticity of the cell states. The problem is that's a whole project, so I've like just kind of been overwhelmed and unable to address these things. But yeah, I think it's super interesting.